So let's go to the next talk. Karthik Gangavarapu was a graduate student in my lab and is now a postdoc with Mark Shushat at UCLA, and he'll be talking about Outbreak.info, which is a good example of how we integrate the mutations, trying to understand the outcome of disease. Karthik.
Thank you, Kristian. So today I'm going to talk about Outbreak.info, really a tool to assess the impact of mutations on viral phenotypes in the context of the SARS-CoV-2 pandemic. Since its first detection in December 2019, SARS-CoV-2 has continued to evolve, and you see the emergence of lineages which share mutations and exhibit different phenotypes. The most concerning of these are called variants of concern that exhibit higher growth rates compared to other variants because they exhibit properties like increased transmissibility or varying degrees of immune evasion. The most significant of these is Delta, which is currently most prevalent worldwide. In addition, you also have many variants of interest that are continuously being monitored to see if they acquire mutations that could also lead to an increase in their growth rates. Now, we've been able to really track the pandemic and the genetic diversity of SARS-CoV-2 closely because of sequencing efforts worldwide. As of October 2021, over 4 million genomes have been deposited on GISAID. The availability of so much data means that we can really track the growth rate of lineages in close to near real time. Now, the growth rate of a lineage is really a function of two factors, epidemiology and inherent biological properties. One extreme example where epidemiology influences the growth rate of a lineage is the B1177 lineage that increased in prevalence in the summer of 2020 in Europe. Initially, it was thought to be because it was inherently more transmissible, but turns out it was only because of an increase in mobility due to travel during the summer. Another example is the variant of concern B117, which was shown to be 40 to 50% inherently more transmissible compared to previously circulating lineages. Now, when we break this down even further, we see that epidemiological factors like mask usage or travel vary over time and differ between different regions globally. When we think about inherent biological properties, it's primarily a function of the mutations found in the lineage. Now, Outbreak.info regularly ingests all the data present in GISAID, which currently is over 4 million genomes, and helps you explore this genomic data across 3 dimensions: time, geography, and mutations. I'm going to show you a quick demo now. This is the homepage. We built different tools to explore different aspects of the genomic data. On the variant reports homepage, we've listed the variants of concern, alpha, beta, gamma, delta. On the right here, we also show the prevalence of different mutations in the spike genes. We also have variants of interest and other variants that are being monitored by organizations globally. You can select either a lineage or a combination of lineage and mutations, or just mutations, and view the variant report for that particular query. Here I'm looking up the variant report for Delta. You see that there's over 1.8 million sequences of Delta deposited on GISAID. On the left, you see all the sublineages that fall under the Delta umbrella. Here, we're showing you the change in prevalence of Delta globally. You can also select a country like the US, and you see a similar trend where Delta outcompeted all previously circulating lineages. Here, we're showing you the changing temporal prevalence of various sublineages that fall under Delta. You can also select a state like California, which shows very similar trends to what we see across the US. Scrolling down further, we show you a breakdown by different counties in California. And you can also look up a location report, which shows you the currently circulating lineages in a given location. Here, you see the most prevalent lineages in the last 60 days in California. We also show you the prevalence of different mutations across all these lineages. Since Delta is the most prevalent, you see that P681R and L452R are most prevalent right now. In the tool below, it lets you actually compare the changing proportions of each lineage over time. We see that alpha and gamma sort of were increasing in prevalence until the introduction of Delta, which outcompeted both those lineages. In addition, you can also compare the changing prevalence of these lineages to reported cases within a given location. Now, because we're able to actually explore the genomic data in these dimensions, Outbreak.info is really useful in tracking the change of different lineages during the pandemic. One example of this is AY.4.2, which was recently found to be increasing in prevalence in the UK, leading to many people taking a close look at this lineage. This lineage in particular is described by 2 mutations in the spike gene. When we look at the prevalence of this lineage in the UK, we see that it's growing quite rapidly, showing that it might have a small transmission advantage over other sublineages of Delta. However, when we look at the same lineage in the US, we see that it was only last detected August 24th, and so it still remains to be seen if the same increase in growth rate is exhibited in the US as well. In addition to tracking the pandemic, Outbreak.info is a useful hypothesis generation tool and can measure things like convergent evolution. One primary example of this is the N501Y mutation, which is shown to increase the transmissibility of a given phenotype by increasing the binding to the ACE2 receptor. When we look at Delta, we see that the P681R mutation is very highly prevalent in this lineage, which is thought to encode for a more optimal furin cleavage site, thereby increasing the pathogenicity of the virus. However, this mutation is also found in the P.1.8 lineage, which is sort of independently acquired. But you can see that there's 2 other mutations found in the Delta lineage that are not found in this lineage, sort of pointing to other mutations that might have an impact on a phenotype like increased transmissibility. Since its launch, we've had over 2.5 million page views, and we've been recently well adopted by the community, including researchers, public health officials, and scientific journalists. All the code that we use in Outbreak.info is public and available on GitHub at these links. Finally, I want to thank the entire Outbreak.info team. They've been working continuously to keep the data up to date so it can be a useful tool. In particular, I want to thank Laura Hughes, who's been co-leading the project with me. Thank you.
Awesome. Thanks, Karthik. Great talk.